Hey everybody, today we are going to do a video on the immune system. And this is a functional system, and what we mean by that is all the cells and tissues are united not by how they look or where they are, but by what they do. And the big function of the immune system, of course, is the ability to resist disease, which is always occurring, nonstop, never takes a break. And in our discussion, we're going to see there are two big branches of this system. There is the innate system, also known as the non-specific system, and then the adaptive system, also known as the specific system. So those are the two branches that are continually resisting incursion and our invasion of our bodies by all sorts of microbial nasty things all the time. All right, so our innate or non-specific immune system, it's going to recognize and destroy foreign things, but the response does not require any sort of previous exposure. So you don't have to have seen the antigen beforehand, and also you don't improve upon any subsequent exposure. So two things it does not do. It doesn't require previous exposure. It does not improve upon subsequent exposure. So all it does is recognizes things as being foreign and kills them, or tries to kill them at least. And so as we discuss our innate or non-specific immune system, we're going to talk about barriers, cells, and chemicals. Now the specific or adaptive system, on the other hand, it not only recognizes and destroys foreign things, but it exhibits specificity. What that means is that the response is tailored to the invading organism. The major cell of adaptive immunity is this guy right here, which you remember is a lymphocyte. And this lymphocyte will not respond to any random pathogen, but to a specific particular one, and the response will be specific to that particular pathogen. And the great thing about specificity is specificity leads to memory. So our lymphocytes will know, quote unquote, when they've encountered a pathogen previously. And because of this memory, as we're going to see, there is the ability to improve upon our response in subsequent exposures, where we don't even know that we're being invaded by the same organism that was in there before. All right, so we're going to come to this adaptive specific immunity eventually. Let's go through the innate branch first. And remember, that was barriers, cells, and chemicals. And one of the major barriers, of course, is the skin. It is an awesome, amazing barrier. We know the skin is a stratified epithelium, so multiple layers here. Harder to get through, many layers than just one. And especially the apical cells closer to the surface, the cells there are really getting keratinized. So they are filled with this tough, fibrous protein called keratin, which is really resistant to degradation by microbial enzymes. But that's not all. Our skin is also constantly making sweat. We are always making what's called an insensible amount of perspiration, so it just basically evaporates really quickly. But what it does is it makes our skin surface slightly acidic, and that acidity is going to retard bacterial growth. So got a sweat gland right here, always making sweat. We got our keratinized stratified epithelium, plus we've got sebum. This is a sebaceous gland right here. It is making sebum. We know sebum is our body's natural moisturizer, oily substance, but it also has bactericidal properties. So it kills bacteria. Now, of course, despite this acidic situation, this barrier, I just did not spell bactericide correctly, or maybe I did. Yeah, there we go. Anyway, despite the keratinized epithelium, despite the acidic sweat that we're always making, and the bactericidal oil that sebum is, we still have a huge epidermal population of bacteria, and this is known as our normal flora, the quote unquote good bacteria. And the good thing about good bacteria is they are using up resources, resources that could otherwise be utilized by the bad bacteria, so nutrients, oxygen, water, etc. All right, so that's our skin, which is a super major awesome barrier. Of course, body fluids can be barriers as well. Sweat, we mentioned already, urine, saliva, tears, all help flush pathogens away, all provide an additional level of protection and resistance to entry. But we're not done with barriers, nor body fluids, because those body fluids in question, all of them contain an enzyme called lysozyme. And lysozyme is an antibacterial enzyme. It is an antibacterial enzyme, and it is in your saliva, it is in your digestive and respiratory secretions, mucus, reproductive secretions, it is in tears, it is in all sorts of different things. And it helps reduce our bacteria population in all those areas because of its antimicrobial properties, its ability to destroy bacteria. I was going to go into the whole story of the discovery of that, but we skipped that in lecture, so we're not going to do it in our video either. Okay, more barriers. So remember, we've got four major tracts. Four major tracts are, of course, digestive, I'm just going to abbreviate here, digestive, respiratory, reproductive, and urinary. And all of these tracts are constantly being invaded by microorganisms. And all of these tracts are lined by these things called mucosae. And this is a mucosa right here, lining the intestine here, so the digestive tract. Mucosae are called mucosae because they are these membranes lining the tracts, and they are constantly making mucus. And that mucus can be thicky. I meant to say sticky, but I was thinking about the word thick, which is the next thing I was gonna say. So I ended up saying thicky. I don't even know. Is that a good word? Well, the mucus can be sticky and it can be thick, and both of these things can stop bacteria from moving, can trap bacteria as well. Now, mucus can also be acidic, especially in organs like the stomach. The gastric mucus there is really acidic, but also the vagina mucus there is acidic as well. Now, the mucus from the mucosae also, of course, contains this antimicrobial enzyme called lysozyme, which we were just talking about. Plus, we have normal flora in all of these tracts, within all of these mucosae. And those normal flora, as on our skin, they are competing with, quote unquote, bad bacteria for resources. So we don't always mind the good bacteria. All right, on that note, we're not going to go over every single mucosal membrane, but when we do hit the different organ systems that have them, like digestive, respiratory, urinary, and reproductive, we will come back in detail on those mucosae. I do want to spend a tiny bit of time with the respiratory mucosa. It is prodigious in terms of its mucus production. It makes a ton of mucus. And not only do we have mucus to trap the inhaled pathogens and other microorganisms, particulate matter, the stuff we're always breathing in, but we've got cilia on those respiratory epithelial cells, and the cilia are constantly sweeping the mucus up. It's called the mucociliary escalator, and we're constantly unconsciously swallowing mucus. Maybe about 500 milliliters, like a regular sized water bottle, a day of mucus we unconsciously swallow. And that mucus contains trapped pathogens as well as anything else that we breathed in. Now, of course, other respiratory events that help us get rid of infectious agents, include sneezing, coughing. Those, while annoying, have immune benefits. All right, so we're done with barriers. Let's move on to cells. And we've got a couple different cells we're going to discuss. So one non-specific immune cell we've got, one innate immune cell we got, are the neutrophils, which we've met before. You know this is a neutrophil. We talked about him being a bacterial killer. It secretes antibacterial chemicals. It is a phagocytic cell. It's often referred to as a small phagocyte or a microphage as opposed to a macrophage. Now, these are our first responders. So if we get an cut, if we get a break in our epithelial tissue, these guys are the first ones on the scene. They are quick to arrive, quick to kill, quick to defend. They often end up killing each other as well. Now, another white blood cell, which we met previously, who belongs to this non-specific group, are the eosinophils, which we know are adapted to try to destroy, parasitic worms, multicellular invaders. So their orangish granules are chock full of cytotoxic chemicals that are going to be released upon any multicellular invader. Now, bear in mind, both the neutrophils and eosinophils are part of the innate or non-specific branch of the immune system. So they don't care, the identity. The neutrophil doesn't care what bacterium is invading. The eosinophil doesn't care what, sort of worm is invaded. It is just going to try and kill them because they are foreign. And on the flip side, neither of these cells gets any better the next time we meet the same organism. All right, another cell we've got to be aware of in our non-specific defense are the big eater cells, the macrophages. And we'll look at their mechanism of phagocytosis in a moment. Macrophages, of course, remember when we talked about them in blood, they are derived from blood monocytes. And macrophages are generally categorized as either being free or fixed. The lower picture here is from the lung, and the macrophages in the lungs, and we've got the guys with the M's on them here are macrophages in the lungs. Those macrophages are going to be free macrophages. The idea being the lung has this tremendous surface area that macrophages need to patrol, looking out for any sort of invader. Now, in contrast, fixed macrophages do not move about. They basically stay in place. This is the liver up here. And recall that the liver has sinusoidal capillaries. So we got one here, got one here, got one here. And macrophages, which in the liver are referred to as Kupffer cells sometimes, they just hang out in the walls of these capillaries and watch the blood go by looking for things that need killing. Now, how do macrophages go about that killing? Well, they're macrophages, right? So they're big eaters, and cellular eating, of course, is phagocytosis. So here we've got a pair of pathogens, let's say. The phagocyte is going to grab onto them. It's going to stick out a pair of cellular extensions, and we see the two of them right there. These cellular extensions are called pseudopods. And what happens is the end of the pseudopods are going to join together. The membrane here and the membrane here are going to fuse together. And then you've engulfed the microbes in this phagocytic vesicle, this little membrane-bound bag, which is known as a phagosome. Now what happens next is the phagosome is going to fuse with a small organelle known as a lysosome, which is basically a membrane-bound bag of digestive enzymes. And once those two fuse, we've got a phagolysosome here, and the enzymes are going to chop up and destroy the microbes. And sometimes the microbes, like bits and pieces, stay inside the macrophage, but sometimes, as in this image here, they are going to be exocytosed, just spat out of the other side. Now, of course, bacteria and viruses have their ways to resist like every step of this process, from the engulfment to the fusion to the destruction via the enzymes. Pathogens are doing their darndest to try and stop every step of this. Okay, an important related idea is something called opsonization. An opsonization refers to when an antibody, which is a special immune protein that we're gonna deal with later, maybe in this video. Depends if we do one or two for the immune system. But an antibody is a special immune protein made by B lymphocytes. And when an antibody or this other type of immune protein known as a complement protein, when either of these guys bind to a foreign thing, that increases the likelihood of phagocytosis. So macrophages are much more likely to engulf a bacterium with antibodies attached to its surface. And so similarly over here, macrophages are much more likely to engulf this bacterium if he has an activated complement protein attached to him. Notice we're not drawing to scale here, by the way. The complement protein and the pathogen, the macrophage are not all the right size. Anyhow, the word opsonization literally means to make tasty, and what's being made tasty is the pathogen, the bacterium. What's making it tasty is the antibody or the complement protein. And because they're tastier, the macrophage is more likely to eat it. So when we do talk about antibodies, we will come back to this idea of opsonization. All right, another nonspecific cell that we have is the natural killer cell, which is a special type of lymphocyte. I know we've already mentioned T lymphocytes and B lymphocytes. We're going to go into more detail on them as well. But there's another special type of lymphocyte called the natural killer cell. It attacks and kills infected cells. So like, for example, if a cell is infected with a virus, a natural killer cell will attack and destroy that infected cell. But here's the thing, the natural killer cell does not care what virus did the infecting. The natural killer cell also will destroy tumor cells. That's what's going on in this picture here. The cell with the with the N on it is a natural killer cell, and this guy with the T is the tumor cell. And we'll talk about the method of killing in a moment. But natural killer cells kill tumor cells, but they don't care like what kinds of cancerous proteins are being expressed on the surface of the tumor cell. They don't care what type of tumor cell it is. So they're non-specific in that way. So another category we got to do. So we've done barriers, we've done cells. Another category we got to do are chemical defenses. And there is this group of non-specific immune proteins called interferons. In interferons, there's a whole bunch of different types. There's a whole lot of jobs they do. We're going to keep it pretty simple here. In general, think about interferons as being made by infected cells. And you could think about this infected cell being like, all right, I'm going to die. But by releasing interferons, I can help save other cells. And so interferons are going to have a couple big jobs they do. Let's cover the one that gives them their name first. So interferons, when they stimulate neighboring normal uninfected cells, they cause the normal uninfected cells to make enzymes that interfere with viral reproduction. The fact that they're causing the cell to make enzymes that interfere with viral replication is why they get the name interferons. Now, this is not all they do. This is really important, though. This helps the spread of the virus. I mean, think about it. The virus wants to get inside a body cell, hijack that cell's machinery to make as many copies of the virus as it can. If we can stop that viral replication. So when the virus infects this cell, it can't reproduce itself. We're going to limit the spread of the virus. Interferons also stimulate NK cells. NKs are the ones we just talked about, natural killer cells. Natural killer cells, of course, kill infected cells. They do it either by releasing chemicals called perforins, which punch holes in the infected cell, or by convincing the infected cell to just go ahead and commit suicide, which is called apoptosis. Okay, finally, interferons are going to attract macrophages. Do you remember how one of the characteristics of white blood cells was positive chemotaxis, where white blood cells have the ability to move towards high concentrations of certain chemicals? Well, macrophages are going to do this. Macrophages are attracted to interferons. And if a macrophage is attracted to these interferons, it could run into this infected cell, the one that released the interferons, and then the macrophage could engulf and kill that infected cell, hopefully before viruses have had a chance to fully exploit that cell in terms of producing more viruses. All right, so that is our first example of a non-specific cellular defense. Another one we've got to do is something we touched on before. It's a system of non-specific proteins called the complement system. And it's a whole group of proteins. Look at all these proteins involved over here. We are totally going to memorize this pathway. And as I'm sure you've guessed, I'm just kidding. We're not going to memorize that pathway. But we are going to know there's a group of non-specific immune proteins known as the complement system. They're made by the liver, and they're in your blood, so they're plasma proteins. And normally, they're in your blood in an inactive state. But we're going to activate them to help deal with pathogens. And we can activate them either directly or indirectly. Like they can come into contact with the particular microorganism, or antibodies can activate these complement proteins. Now, what we're really interested in is what activated complement proteins do in terms of how they protect us. And I've got 4 basic functions of activated complement, and my mnemonic device for that is the word coil. So my 4 basic functions of activated complement are chemotaxis, which we just mentioned a moment ago, for interferons, opsonization, which we talked about a few slides ago, inflammation, and lysis. Now, chemotaxis means we are going to attract white blood cells. So the presence of activated complement proteins, which in this case we see it bound to a bacterium, that is going to attract white blood cells, such as a macrophage, like this guy right here. Now, of course, not only do we want to attract the macrophage, but we want to do something that increases the likelihood of phagocytosis. And that's called opsonization. Basically, the complement protein, when it is activated, it exposes portions of it that are very likely to bind to the arms, the pseudopods of the macrophage, and this is likely to get this complex engulfed and destroyed. Okay, so the first C is the first 2 functions, the C and the O, chemotaxis and opsonization. The I in complement functions, in that coil mnemonic device, is for inflammation, because activated complement proteins bind to this guy. You were probably wondering when we were going to talk about him, especially since we covered neutrophils and eosinophils not too long ago. This, of course, is I know you were thinking of basophil. And basophils, when they're stimulated by complement proteins, they release those 2 H chemicals, histamine and heparin. Histamine and heparin are both going to increase capillary permeability and increase local arteriolar vasodilation, which decreases resistance, which increases blood flow. And with more blood flow and with more capillary permeability, we're going to get inflammation. And inflammation is the first step in the healing and the protective process. So don't think of inflammation as a bad thing here. All right, we'll talk more about inflammation in a moment, in fact. Let's go to the L in coil. The L is lysis. And what's going on here is we have a group of 5 different activated complement proteins, and they've all got different names that we're not going to worry about. But when this group of 5 is activated, what they're going to do is assemble into this structure called a membrane attack complex, which is then going to basically punch a hole in the wall of our bacterium or in the envelope of our virus. And when we punch a hole in it, that hole lets sodium rush in, and then osmotically water is going to rush in as well, and the cell is going to swell and burst. And of course, that word for cellular bursting is lysis. So that is the L. Those are the 4 basic functions of activated complement proteins. All right, we're now going to talk about 2 more non-specific immune situations, inflammation and fevers. And then we'll call it quits for immune system video number 1, and we'll save the specific branch for immune system video number 2. So inflammation is a good thing. It's the first step in the healing process. It occurs anytime tissues are damaged. Inflammation is going to prevent and reduce pathogen invasion, help us dispose of broken and damaged cells, and set the stage for repair. Now, there are 4 basic signs of inflammation, like what's going on in an inflamed tissue. And before we get to those 4 let's do those 4 basic signs. Why not? Those 4 basic signs are swelling and redness and heat and pain. And pain often corresponds with loss of function. So these are my 4 basic signs, and we'll come back to these in a moment. Now, inflammation occurs whenever tissues are damaged. So inflammatory chemicals are often released by damaged border cells. So cells that are on the line between our internal and external environments. So, for example, skin cells, or any of those cells lining the tracts, the mucous membrane cells. Like we got some respiratory tract cells right here. If these cells are damaged, they're going to release inflammatory chemicals. Skin cells down here, if they're damaged, they're going to release inflammatory chemicals. But not only do damaged border cells release inflammatory chemicals, but activated immune cells do as well. So what do these inflammatory chemicals do? Well, they're going to increase vessel diameter and increase capillary permeability. Those 2 in combination are going to give me an increase in capillary blood flow and an increase in fluid loss. With more blood flow and fluid loss, well, there's going to be swelling. Inflammatory chemicals also stimulate nearby pain-sensitive neurons, and that pain, that swelling, can lead to loss of function. The increase in blood flow, of course, corresponds to the heat and the redness that we're going to see. And the great thing about the increase in blood flow and the increase in permeability is we get more access to the damaged area for cells that are going to clean up, cells that are going to defend, as well as cells that are going to repair, as well as all the resources that we need to do those things. Okay. So that is our quick run through of inflammation. Let's talk about fever for a second here. Fever is a systemic, so body-wide, increase in body temperature. And what's going on in fever is activated immune cells. So immune cells that are in the fight against microorganisms are releasing chemicals called pyrogens. Pyro means fire, gen means create. So literally these are fire creating, heat creating cells. And what pyrogens do is they act on your hypothalamus. You have a cluster of neurons in your hypothalamus that is known as, not as known as, but is serving as your thermostat. So in response to this increase in the temperature setting, it's like when you bump up the temperature on your thermostat a little bit more. It's going to cause your heat pump to turn on, cause your furnace to turn on, so that heat is generated to bring the temperature of your house up. And this is what's going on in our body. When we bump up that temperature set point, we're going to raise body temperature primarily due to increased muscle contractions, but also due to behavioral adaptations like putting on more layers of clothing, blankets, etc. Now there are two big benefits rather, of fever, of this rise in temperature. Number one, it increases the reproductive speed and the mobility of our own white blood cells. And that's good. We want our white blood cells to be more active and to be reproducing at a greater rate. Now, also, it does the opposite for pathogens. It reduces the ability of pathogens to reproduce. So we got the best of both worlds. We got our cells, our white blood cells, reproducing faster, and the infectious organisms reproducing more slowly. And with that, we are done our non-specific branch of our immune system. We're like exactly halfway done with the slides of the immune system. So we're going to stop at 32 minutes in. Great stopping point. Let me know if you have any questions as always, and I will see you later. Bye-bye.